Alright. So I'm gonna go through and mark off attendance, but like we're gonna wait a little bit to see if people get on. Let's see, I have Addison, I have Chris, I have Keegan, and I have Madison. Do you guys have any questions while we're waiting for everybody else? You guys are way less concerned about Top Hat than the other guys were. It was like a big clamoring. Everybody was trying to figure out. I got on and immediately it was like Top Hat, my Top Hat's not working, my Top Hat's not working. I'm like, I'm aware. We talked about this yesterday. But yeah, I'm working on it. It should, I've done a thing. I can't test it from you guys' side. So I did a thing that I'm hoping allows it to link to your accounts. Because I've been talking with IT for Top Hat. So if you guys get a chance to try it and see if you can link it, let me know. But go through the course materials thing. It has to be so complicated. I have Mark Carter. Got 4 other people potentially that we're waiting on. And I won't wait forever, don't worry. Let's share my screen so you don't have to like look at me directly the whole time. Let's see. I guess while we're waiting, I mentioned a little bit about like my past or whatever the last time, but just so that you guys get it, I really prefer focusing on pathogens, the disease causing microbes. Because what I've studied, when I was getting my master's degree, I was working on my PhD. I went to the University of Pennsylvania. It's in Philadelphia. Um, and I was, pretty successful. I was doing really well, to be clear, so successful. I was studying it's an Ivy League university. It's actually founded by Ben Franklin and one of the founders anyways. And it's ridiculous, right? Before the US was the US. And whatever, so it's cool, right? Super cool university. I worked with the CDC as well as the Army because the viruses that I studied were deadly viruses like Ebola and Crimean Congo hemorrhagic fever virus, it's a hemorrhagic fever, Rift Valley fever, and some other ones. But that's what I was studying. I was doing very well, and I got married, and my husband did not want to move to Philadelphia, and he said that so this was after we got married. Less than a week after I moved back to Philadelphia, he let me know that he was not moving to Philadelphia, even though I had gotten a new apartment and all of this, and I couldn't afford that by myself. So I quit grad school for him. And then I moved to Midwest City where he was living his dream of being an engineer at Tinker Air Force Base, which I guess trumps my dream, but whatever. I'm not married to that guy anymore. But that's kind of my story. But cool little side to that. The person who I studied immunology, you probably know, but just in case you don't, it's what deals with how your body fights off infections. I studied immunology with a man named Drew Weissman at the University of Pennsylvania. I know he's my instructor, and that's where I learned from. And he just won the Nobel Prize for the COVID vaccine. So thought that was pretty cool. I get to, brag about another thing now. But yeah. So, all right, that's enough blabbing. But just so you guys know, that's kind of what my background is, and that's kind of why that's what my background is, and that's kind of why I want to study on the pathogens, which I think is what you care about too. So it works out. But it turns out I have to teach you about the other crap too. I'm sorry. But let's talk about the lab first, shall we? Let's get into that. Lab safety. Before we get into it, just once more, guys. Anybody have any questions before we get started? Because we got some more people that joined in. All right. You guys are probably familiar with a lot of lab safety rules, so it's probably nothing new. Just be sure you read through the lab safety rules.
Good idea. We're gonna get this started with the drink.
And anyway, so that microbiology lab safety list that's there, just be sure that you look through that, right? We're going to talk through it here right now, but you have to open it anyway to get to the other stuff in the class. So, well, 100% required lab coat and closed-toed shoe. Crocs are not closed-toe, they have holes in it. So if you were to spill anything on your closed-toed shoe, that would soak into those little holes. If you're wearing a tennis shoe, could it soak into that too? Oh yeah, of course. I mean, yeah, but it might give you a chance to get your foot out of it before it started soaking in, right? So I don't know, but them's the rules. You might be asked to leave. You might be counted as absent because you were asked to leave. It says you will receive a 0. Okay, listen, if you can go and get shoes from somebody, a friend, Walmart, I don't care. Or you can get your lab coat from somebody, wherever, I don't care. Then come and join the lab, right? But if you can't, or you just choose not to, then yeah, you're going to get a 0 for that day. I'm not going to let you get away with a whole day of not working and then come back and then be like, oh, I bet I didn't have the lab coat. Why are you counting me off? Like I couldn't have participated. Yeah, let's all not wear lab coats and never go to lab. That sounds, I would do it. Like it's got me out of it. But that's not how that's going to work, right? So you will get penalized for that. But if you can take care of that problem and get back to lab and you can actually still finish your work, then I won't count it against you. I'm not very picky about being on time or being late or whatever. I say, let's see. No, you can't keep your lab coats in the lab because we have other students that are going to come in and out of that lab, right? So that's the problem with it. We just don't have the space for it. In the summer, I would say maybe like that would be okay, but like I don't know where I would have you keep them. That's the problem. That's a good question. If it was just us using that lab, you guys are not the only class that uses it, even in the summer. So I have 2 sections. So there's a section right before you guys, and they have more students than you guys do. If it wasn't the case, if it was just the one section, then I wouldn't mind it. But I just don't know, like if you have 2 lab coats in a cubby and the other person takes the other person's lab coat and now somebody doesn't have a lab coat, and just be responsible for your lab coat. If you get something spilled on it and you need to take it home and wash it, and you don't want to carry it or put it in your bag or whatever, just let me know. We'll get you like a biohazard bag or something to put it in if we have to. That's only happened once. So just let me know if that does happen. Or we have a we did, I don't know anymore, but we used to have a washing machine here on campus that I could use for it, but it's weird. Things are weird right now. Okay, that's basically what it is. Things are real weird. Oh, for those of you who came in late, that reminds me of weird stuff. Speaking of that, top hat. If you guys want to try that out for me, I think I've got it linked. I really tried to get it linked properly. I've been talking with the IT guy and been trying to work on that. It's just I can't test it from my end and I can't see what it looks like on your end. So if you guys do have time to check it out, let me know if you're able to access it. Awesome. Thank you so much. So for those of you that aren't in the chat right now, Carlos just said that it seems to be working. Yeah, okay, good. So I'm glad that that's connecting and working out for everybody, at least so far. But yeah, we'll work through that. I'm pretty lenient about stuff like this. I think that's why the other class was freaking out. They just decided like, well, how much is to get my work done? How much do, I can only, the thing is due on, it says it's due on, geez, guys, like if it says it's due tomorrow, by 10 a.m. or whatever, and you don't have access to Top Hat, I'm not going to give you zeros. Please. Whose fault is that? It's not yours. So just, it's okay. But it is working now. So I guess no excuses. If you really can't get your Top Hat working, try to keep in contact with me or Top Hat, depending on your issues. But yeah, we're going to assume everybody can get the pre-lab done before tomorrow. So a heads up. And I'm not super picky about it right now, just simply because we're not even in the friggin' lab. So that's where it's really going to be more important, because I see it as a safety issue. You come to lab prepared, so you know what you're kind of getting into, you have an idea at least. Otherwise, you could be a risk to the people you're working with. That's the idea. I mean, it's kind of extreme to put it that way, but yeah, come prepared. Personal items, drinks, and snacks. You're welcome to leave your backpacks. Typically, I'd say leave your backpacks and stuff like that out in the hallway, because nobody typically uses those other rooms when we're using them. So we're the only ones in that area. But we're having weird construction. I don't even know what's going on in the building, honestly, right now. Like, honestly, and neither does Dr. Rhodes. And so it's just like we don't now have any idea what's going on. Normally. Normally, I'd say you could leave your stuff in the hallway. You could potentially bring it into the lab. What I'll say is if we have to, and you have drinks or something like that, that we don't want that at the bench where you're actually working with live bacteria, right? Bacterial cultures of E. coli, of salmonella, and all these other things. They are non-pathogenic, but that doesn't mean they stay that way. Bacteria can pick up genes from other bacteria that they are related to in the environment, including the bacteria on your body. And if you think you don't have E. coli on your body, I hate to break it to you, y'all. Y'all got all the E. coli. So, you probably have weird strains too. It's just how it goes. So keep that in mind. I don't know. Anyways, keep your drinks away from it and keep stuff out of your mouth in the lab. Yeah, that's all gross stuff. So don't do it. Be conscious of that stuff. If we can't leave our drinks out in the hallway, which we might not be able to because of construction, but you have drinks, try not to bring them into the building. But if you do, then just let me know and we'll put them somewhere in the lab where we're not dealing with live cultures. Okay, we'll just do it that way, like in a cabinet or something. Okay, so just let me know. I know what happens. We have our drinks sometimes. I have two right here, right now, so I can't judge. So we were talking about oral transmission wounds. If you cut yourself at all, we need to bandage any open wounds so that the stuff that we're working with doesn't get inside of your body, because gross, right? What is going on? Oh, sorry. My sister's cat had to be taken to the hospital because he has really bad diabetes symptoms right now. It's not a good sign. He's kind of older, too. But sad fixes for the kitty. Oliver is his name. Anyways, if you have an open wound and you come to class with an open wound, let's say you were weed eating and you cut yourself on your hand, and we're working with our hands with live bacteria, let me know. I have some gloves I can get for you. We typically don't wear gloves in class. If you want to wear gloves, bring your own gloves, please. I just don't have enough for everybody every day. I just can't provide that. But if you want to, you're welcome to. Otherwise, if you injure yourself, let me know so that I can get you some gloves and we can bandage your wound. so that it's not open to any spills or anything that you could encounter in the lab. That's what that is about. And if you do get a wound in the lab, please notify me immediately. You'll notify me about any accident that's in the lab. But yeah, if you get a new tattoo, if you get a new piercing, please do not touch it in the lab. If you think you did and you're like worried about it and you're not sure, let's say you get a tattoo and you're worried that you splashed it on you're splashing stuff on your arm. You're like, I just got it done like 2 days ago, whatever. I got you. Okay. I got you. So just let me know if you do think that that happened, something like that, because that we consider that to be, if you guys don't know, that's an open wound for quite a while after. Piercings is the same sort of thing. They take a while to heal. So if you think you touched it just absentmindedly or something, I have some fragrance-free sensitive skin soap that I use on my own tattoos and piercings, and we can wash it with that. Hopefully we can get it taken care of, and then you can go take care of it at home and you are less likely to have issues with it. Let me know. It's all okay. Keep your environment clean. Come into lab, clean your bench top. Just assume that the person who was there before you was disgusting. Okay. Clean your bench top when you are leaving. Just assume that everything that you worked with has little microscopic droplets everywhere that you were. Clean your stuff for the next person, right? Keep it cleaner than you found it. If it was a hot mess with all your bottles all over the place and spilled and stuff in the sink and whatever, I mean, organize it. You don't have to be perfect with it, but organize the best you can for safety and for cleanliness. Okay. It doesn't hurt to clean your dry erase markers. We will be having those clear envelopes that I'll insert your worksheet into, and you will write with dry erase markers on it, take a picture of it, and then upload it on a top hat. That's how it is going to go eventually. But it doesn't hurt to clean those either. They're not required to clean it. Please clean your bench tops. If I don't see you cleaning, if you aren't cleaning those regularly, I might get onto you. Okay, fire. We use fire almost every day. Bunsen burners, literally almost every single day in lab. Microscopes is about half the class. Fire every single day. Okay. There are fire extinguishers in the lab. We'll go over that when we get into the lab. There is an emergency gas shutoff near the door. There's only one door in the lab. So just hit that button if there is something serious going on. Long hair. This is sort of, I feel like medium hair, but that doesn't mean you get away with it, right? If your hair is leaning forward at all, like while you're one of those people that leans a lot and like really focuses in like I do, then maybe put your hair back when you're in micro, okay? If your hair is actually long, tuck it into your lab coat. If you don't have a hair tie, that's fine. At least do that, okay? But I prefer a hair tie. Just put it up before you come to lab, okay? Yes, this is actual risk. Please do not, I don't want to deal with that ever again, okay? If you don't, it's not that it's people get hurt, it's just people freak out and there's just big old, like I don't know. It's just not do it. If you don't know how to safely operate the Bunsen burner, ask for help or a demonstration or something. Yes, here's the thing, okay? We're going to get to this in a minute because this is a thing I can tirade about. But if you have people sitting at your bench with you and one of them knows how to use a Bunsen burner and they want to show you how to do it, that is fine. That is fine. I have no problem with that, okay? Help each other out. If you're competent in how to use it, it's fine, okay? Never operate microscopes if there is a lit Bunsen burner on your bench. We don't want that flame accidentally melting through the power cord, or you're not paying attention because you're looking at the microscope and now you burnt your hand or something like that, right? So if you get a microscope out, the entire bench needs to have its Bunsen burners off, okay? Disposing of materials is pretty straightforward. If you had a Walmart bag, you have an item that you need to get rid of. Let's say a glass slide. I don't have any in my office because apparently we're prohibited from anything in the lab. So glass slide, we'll be making quite a lot of those. And they're just sort of sharp, jagged edges. They're made of glass. Even if it's not broken though, if you fill an entire like Walmart grocery bag with just those glass slides and tie it tight to get the air out and whatever, what are the odds that one of those edges is going to poke through the side of that plastic bag? Pretty high, right? If it's sharp enough to do that, then put it in the sharps container, okay? Because if you put it in the biohazard waste, it will poke through the biohazard bag and potentially leak all the liquids and nasty that's in that bag out. So don't put anything remotely possibly sharp into the biohazard trash, okay? into the sharps instead, even if it's not broken. Obviously, broken glass is going to go in the sharps container. For the biohazard waste, it's anything that is disposable that has touched bacteria. So if you have a paper towel that you cleaned up a spill of a culture, like there's a whole bunch of bacteria growing in a broth, and you spill it, even just a little bit, and you clean it up with that paper towel, that's now contaminated biotrash, right? But if you came into lab and you cleaned your desktop with a regular paper towel with regular disinfectant, there's no spill or anything, you clean that regular trash, it's not contaminated, okay? So think of it as if you would, it's a throwawayable item, a little plasticky pipette, swabs, like cotton swabs that we use, and we'll sometimes do samples with. The Petri dishes, those are those round plastic things. They're just made of plastic. They're disposable. Anything like that that had bacteria on it goes into the biotrash. If you never put the bacteria on it, but let's say, I don't know, you dropped it and it got dinged up all of the agar that you're going to grow this is solid surface for your bacteria or whatever. You just don't want to use it, but it doesn't have anything that you put bacteria on it directly. That could go in the regular trash, okay? If you're ever not sure, just ask me. Test tubes themselves will be growing a lot of samples in test tubes, and you'll probably have a little way to label it with some tape. Take the tape off before you put it on the disposable cart. So if you have these glass test tubes that we will reuse and wash and everything, we will sterilize them using an autoclave. An autoclave is a machine that uses steam and heat and pressure to sterilize. Sterilize means we're removing all life from it, okay? There's no traces. If you boil something to treat, like, let's say, milk bottles for a baby or something like that, that does not sterilize, okay? Don't just go camping and then boil your water and call it sterile. That is incorrect. Some things can survive that. A lot more than you would think, okay? We'll talk about what throughout the class. No worries. You get to learn all about that. But autoclaves do sterilize, and so that's why we use them and not just boiling. So we will sterilize your tubes with an autoclave once we have enough worth our effort, right? We'll collect them on a little rack and then take them to the autoclave and go get them sterilized. But the tape is really hard to get off if it goes through the autoclave, and we don't necessarily have time to go through everybody's all at once. So please just do yours, just yours, okay? Take the tape off. Otherwise, your tape is going to go on what we call the wall of shame. And it's kind of what it sounds like, okay? All righty, and then you get me frustrated after a while. Okay, that's that. I'm pretty happy with that. Yep, if you don't know where it goes, just ask. Big on that. I'm really big on asking, and we're going to get to that in a moment. Oh my gosh, sometimes things they say on the email, BS, spam emails. Sometimes the titles are things that make you be like, my car is what? But it's not. It's a fake lie. It's a lie. Accidents of any kind need to be reported. If you break something, tell me. If you spill something, tell me. If you got cut or you have an open wound or whatever it is, tell me. If somebody's calling somebody names and you think it's completely inappropriate, tell me. I think one of the things to find is I'm pretty chill and I want to help everybody out, and everybody mistakes that for just like super nice, friendly, you can get away with anything until you see my backbone and you don't want to. So don't put me in that mood. So just let's all get along and be honest about things. If you're trying to hide any sort of like accident, like I was just telling the other students before you guys, if you, like, let's say at the exam, this actually happened. I had a student who was looking at one of the tubes that had liquid in it. It had bacteria in it, in the liquid, right? And she was tipping it, and sometimes they will solidify and sometimes they won't. So she was just seeing if it was solid or not. But she's tipped it so far, which is something I warned about before the class. And I said, if I catch anybody did this, like, it could cause you points on the question associated with that too, because you should know by now not to do it as the final, right? Don't tip stuff and spill it. Geez, you already made it through a whole semester and you're still spilling things. So she had tipped it. The caps are not watertight. They're not airtight. They're actually made so they can get air into the tube. And she spilled it all over the desktop. I didn't discover this until later. And then it was one of the other students that told me that it was this particular individual. Since I didn't catch it happening, It's not really fair. You can just go by words as other people. That's not fair, right? So I understand it, but she did. She spilled this culture all over this desktop, and she just left it there. Other students being who they are, they just like answered the question around it and didn't touch it. But like, imagine you went over there in your flustered test taking state and you put your hand in a bunch of E. coli. Like, gross, right? Like, So if you spill something, just like raise your hand and be like, Professor Yost, I know you said not to spill anything, but I accidentally spilled this. And I'll be like, oh, okay. Well, do you know how to clean it up? Or I'll come over there to help you clean it up or whatever it is. I usually ask you if you know how to clean it up appropriately. And if you say you do, I'll let you clean it up. Like, it's not hard for the most part. You wipe it up with a towel, paper towel, just the nasty, put it in the biohazard. It's contaminated. Then get it with some disinfectant and clean that off. It's not contaminated. It had disinfectant on it, regular trash. And that's it. If you're not sure what to do with the tube because it's all nasty too, then you can ask me. It's fine. I'll help you. Always ask. I am there to help you. Okay. But yeah, do not hide it from me. It will not be better. It will not be better. So spills, just let me know. I'll usually ask you if you know how to clean it up. Yeah, I got it. And I'll be like, okay, cool. Or if you want help, just let me know. And I will happily come and help you. No joke. Broken glass without any injury. You just accidentally dropped your tube and it shattered. And yeah, there's probably broth everywhere and we'll need to clean it up, but that's fine. But these things happen. It's an accident. You didn't do it on purpose. So I'm not mad. So you can call me over and I will help you find all of the glass, make sure we get all of it, make sure we clean up all the liquid so there's no more thing. That's part of why you don't want to be hiding things from me. Because if you hide it and you didn't clean it up properly, you will leave a hazard for other people. And that is so disrespectful to your classmates. So please just, if you make an accident, just let me know. Okay. If somebody gets hurt from whatever your accident is, maybe somebody slipped, somebody broke, glass and cut their hand or whatever it is, or grabbed one of the fiery hot loops that we use and just wasn't paying attention, something like that, right? It gets pretty hot. I say it gets pretty hot. It sterilizes. It's that hot. Okay. So don't touch it. But let's say you did hurt yourself, just let me know and we will treat it with the first aid kit where we can. And if we can't, then we will call 911 or whatever appropriately, right? So make sure that we can take care of you. That's what this is about you being safe. Always practice aseptic technique. We'll learn about what that is and how to do that in the next lab, but it basically has to deal with keeping things from contaminating. So if this is your sample, this fidget spinner, let's say, and I use fidget spinners a lot, sorry. If this is your sample here, you're trying to keep stuff from getting into it, right? So it doesn't contaminate your thing, but you're also trying to keep your thing from contaminating the outside. So it's both ways, but minimizing contamination, right? That's the goal with aseptic technique. So we want to always practice that and be aware of that. Wash your hands whenever you leave the lab. If you get stuff on your hands during the lab, like some of the liquid or whatever it is, wash your hands immediately, please, and then come back and do your work. But it's not going to hurt you if you just get it on your normal intact skin, okay? But please wash your hands. Don't take it home, right? Don't drink the ethanol. Our ethanol, it is drinking alcohol, but it has methanol added to it so that we don't have to pay for a liquor license. If you have just ethanol and water and that's it in it, that is technically of drinking grade, and you have to pay for a liquor license in the state of Oklahoma, even if you're using it for scientific purposes. So we have a version of it with methanol in it. Don't drink it because it's made to make it non-consumable. Yeah, and it's not because it would make you go blind. So don't drink it. Don't operate Bunsen burners when I'm not around, especially in the beginning, first few days when we finally get started with Bunsen burners. But at some point there might be a situation where I need to go up to my office to grab something or whatever, just briefly. And if you've been using Bunsen burners without incident for the last few days, I might leave you for a few minutes, but I try not to leave the lab. It makes me uncomfortable. If you're on immunosuppressants or you have an immune disorder, something that makes it hard for your body to fight off infections, including pregnancy, just let me know so that I'm aware, and be sure that your doctor has told you that it's safe for you to participate in the lab. We use non-pathogenic strains. It shouldn't be a problem for most people, but I want you to be safe. So please, it's not for me to decide. It's for you and your doctor to decide. If you have any questions at any point ever in the course, please do not hesitate to ask about anything. Please, I just much rather you have a better sense of what's going on around you than to just be feeling like you're lost. And maybe it sounds mean, but I will not coddle you. Okay, what I mean by that is, let's say you're at a lab bench of 4 people, right? 3 people get along pretty well. They know each other. And the 4th one is just somebody that just sat there, but they also tend to be a pretty quiet person. And the other 3 are working along pretty well with one another. The 4th person has no idea what's going on and isn't able to really even comprehend what they're supposed to be doing. So they just sit there most days in the lab. You think this, I mean, I know you guys know this happens. Yes. This is how some people are and how they get through the lab, and they'll skate by on the success of their partners. They're not meaning to be like that. They just don't know what they're doing, and they're just kind of like confused the whole time, and they never ask for help. And they're just like, okay, I guess I just don't know what's going on. I watch my labs, and you sometimes will think that I'm not paying attention, but I listen pretty much to what everybody's saying all the time, just hint, hint. If some people think I'm not listening, and I'll hear them like talking about me, it's whatever. I never take it personally. I really don't. But seriously, um, I am watching as well. So if you are one of those people who just has no idea what's going on and you have a really hard time asking for help and you're hoping that like this isn't one of those classes and then I'll come around and help you, I will not come around and help you. I will let you drown intentionally on purpose, because most of my students are going into nursing. And if you can't even ask me for help, what are you going to do with your patient whenever you need to ask the charge nurse or the doctor for help? You don't know what to do. You don't deserve to be in nursing if you can't ask for help. So it's time to get over that. And so this is the moment. I will leave you there. If you don't ask for questions, I'm gonna assume you know what you're doing. Occasionally, if I think that somebody doesn't know what they're doing and they're trying to figure it out genuinely, and they're still not very good about asking the questions though, then I'll come over and be like, do you need any help? And then it's up to you to be like, yes, I do. Can you help me with this? But if you still say you're like, I'm fine, then I'm gonna let you be fine. Okay. So that we're clear. Yeah. So if you have a question, please speak up. I see anybody who is able to come and ask a question from the professor, I see that as a strength and not a weakness. We all get nervous when we have to go to someone who's superior, quote unquote, to us, someone who, we see as a higher up, and we have to go and admit that we did something wrong or that we don't understand something that they already explained or whatever. Please, like, I understand that that's life. That is real life. Please come to me if you have questions. Okay. I am aware of how life works, believe me. I'm not gonna poo-poo on you for that. Be sure you watch the lab safety video. It's about 20 minutes. Watch it on double speed or something. Okay, we're gonna move on to ubiquity of microbes. All right, microbiology. As much as I wish it were all about just the pathogens, and pathogens, by the way, are just disease-causing microorganisms. There's microbes everywhere, and not all of them cause disease. And I'm sure you guys know this. There are plenty of microbes that like are totally tame normally, but sometimes cause disease in certain circumstances, right? So we're going to go over a few of them. You don't have to memorize these organisms as we're going through them. These are just examples. We'll go over them individually later on in the course when you're more familiar with the concepts. But we're going to introduce just a touch of it. Today, like I said, you're not going to get tested over it. But there can be microbes pretty much everywhere. Living beings inside of you, on you, right? Not just you, but other creatures. So trees even. Soil, water, and air. And yes, and that's a little complicated, but we'll get there. All right, on or in living beings, there's so many organisms, but here is a list of a few. Streptococcus pyogenes, Streptococcus pneumoniae, Streptococcus mutans, Propionibacterium acnes, Staphylococcus aureus, Helicobacter pylori, Escherichia coli, Klebsiella pneumoniae, Candida albicans, Candida auris, Neisseria meningitidis, and Clostridium difficile. All of these are organisms that can live tamely, normally, healthily, expectedly on people's or in people's bodies in various areas, right? When they get overgrown, when they get out of the area they belong, that's the problem. So all of these can be normal bacteria that are found in people's biomes, normal, what we call the microbiome, right? Like whatever's in your gut or on your skin, we have them everywhere, right? We're learning about our gut biome all the time. It's a very serious thing right now in science, how important it is for your health and not just for your gut health, right? For your overall health. Oh my gosh, there's so many ties to your mental health with your gut. That's nuts. But we're not going to get into that right now. So that microbiome, you have microbes in your gut, obvi. You have them on your skin, obvi. You have them that just live like in your sinuses or in your mouth or whatever, obvi, right? All of these guys can be part of your normal microbiome. All of these guys can also be pathogens. Some of these you probably heard of, right? So you probably heard of C. diff. C. diff is a bacterium. It causes pseudomembranous colitis. That's the name of the disease that it causes. And with pseudomembranous colitis, basically it can cause like a membrane of the bacterial growth in the colon that can cause issues with the movement of fecal matter through the colon and all, whatever, but also inflammation of the colon itself. And it's just a gross smell and it's diarrhea and it's mucousy and gross. So that's C. diff. It's easy to get it spread throughout a hospital. Once you have one person with C. diff, it's like everybody does. And that's because C. diff is a bacteria that produces endospores. And we'll talk about what those are in chapter 4, if you haven't already got to it, which I imagine you haven't. That's fine. But endospores are protective structures only some kinds of bacteria can make. Clostridium species, they can do it. And so it survives pretty much anything. So you can have normal a whole stadium full of different bacteria all over the place that all helping out and working together and taking up all those stadium seats to prevent bad guys from sitting down in it, right? And maybe a couple of those are C. diff naturally. You take an antibiotic because you got knee surgery, you cleared out the stadium seats, except for C. diff. And then what happens? It overgrows. And then you have C. diff, as they would say in the hospitals, but pseudomembranous colitis. And so that's the idea. So that's where it comes from. It's typically from inside that patient's own body. And then it can get transferred to other patients in the hospital because endospores can survive anything. So you think you're washing it off? Honey. You think you're getting rid of it whenever you sterilized that? You probably didn't sterilize it like you think you did. So that's the issue. It survives pretty much anything. Not only is it all quite like that, let's pick a good one. So this one here, Neisseria meningitidis, it sort of gives away what it causes. So Neisseria meningitidis, this causes meningitis. So you've got your sore neck, your stiff neck, you've got your fever, you've got sensitivity to light, and you've got fatigue and all of this stuff, right? And you've got a rash on your body, a petechiae rash. It's like little spots all over. You have meningitis, and not just any meningitis. You have meningitis caused by Neisseria meningitidis. That's the name of the bacterium. And that particular type of meningitis is the most deadly kind that you can have. But what happens if you have meningitis and there's no rash? Because that one always causes a rash. It's the number one cause of meningitis, bacterial meningitis, is going to be Streptococcus pneumoniae. Yes, it also causes pneumonia. But this also causes meningitis. So this is the pneumococcus. You probably heard of it. But both of these are normal in your body, and they're meningitis. Bacterial meningitis is deadly. But Neisseria meningitis can live in like your sinus area in small amounts. Streptococcus pneumoniae, it can be anywhere kind of in your respiratory area, typically in the upper respiratory area, as long as it isn't overgrowing and isn't down deep in your lungs, then and isn't in your friggin' meninges, then you're probably fine. But yeah, man, isn't that cool? A grand majority, I think it's 80% of all UTIs are caused by E. coli. Urinary tract infection, ever had one? Probably, if you're a female. It came from your E. coli from your own booty. Surprise. That's where your E. coli comes from. It's not from somebody else. We can go into how that works later. Well, we've all been there. So, anyways, so Yeah, it could just be stuff that's normal, but it then is a pathogen in certain circumstances. So cool. So plants rely on bacteria in general to turn always, this is an always circumstance, to turn nitrogen in the soil into any form, into a form that they can actually use. Why do you need nitrogen? You need it to make DNA and RNA, right? Those nitrogenous bases that are involved in the nucleotides. Those are nitrogen, and so are amino acids. And amino is an NH2 chemical group. I told you guys you're going to need chemistry for this course. So anyways, you need those. And maybe you're thinking like, man, but you really need, because you really need DNA. You really need RNA. You really need protein, y'all. Protein does everything in your cells. Every single gene in your cell either controls the production of another gene or it makes protein. Nothing else. It doesn't do anything else. It doesn't make anything else. Maybe RNA, like structural RNA, like for ribosomes and stuff, but like everything else. That's for making protein, by the way. Protein, man, it rules us all. How do you think your cells get put together? How do you think your cells get organized? How do you think your cells are directed and whenever they're dividing and things like that? Proteins do all of that. So, man, do you need proteins? And nitrogen is important in amino acids that are the building blocks for proteins. So, so we need nitrogen. So we need it. So here's some bacteria that are found in the soil. Yay. These guys here, you can actually see them present in your probiotics. They're pretty common there. There's other ones too, probably, but cool. And then there's these guys that are also very commonly found in the soil. And I mean quite commonly. Bacillus anthracis, anthrax, Clostridium tetani, tetanus, Clostridium botulinum, botulism, Clostridium perfringens, gas gangrene, Bacillus cereus, causes food poisoning, Mycobacterium leprae, leprosy, Pseudomonas aeruginosa, skin and nail infections, Leptospira interrogans causes lepto. You might be thinking of animals, and of course, animals can be affected by this, but it can also infect humans, and it can be deadly. It can cause neurological issues like meningitis and stuff. Toxoplasma gondii rules the world, I say all the time. It's kind of a joke, but if you don't know, it's the little protozoan guy that mice get that makes them more risk-taking, so that cats are more likely to be able to catch them. They'll go in front of the cats and stuff like that. And people get it all the time. And it also can have an effect on human behavior. This was the one why you don't touch kitty litter whenever you're pregnant, because it can be quite serious to the fetus. It can be abortive. So let's not touch kitty litter. Enterobius vermicularis. This is pinworms. These are the ones that leave little kids with the itchy booty. And you put tape on, I'm not kidding, put tape on the kid's anus and then check it the next day for worms and stuff. And you can even buy over-the-counter treatment for this. It's not uncommon, truly. So, but yeah, that's a worm infection. Then we have Coccidioides immitis. This causes valley fever, and that can lead eventually to meningitis. It's a fungal meningitis. So, fun. This is the stuff that's normally in your soil. Not everywhere, but that's where it comes from. All right, water. In the marine area, we're having currently right now, fastest spread of diseases amongst marine wildlife ever recorded in history. This is actually related to global warming because we're having blooms of these bacteria and stuff, and then them infecting the wildlife. Sorry, it's emails. Vibrio vulnificus and Vibrio parahemolyticus. I don't see those ones a lot, so I had to think through them. But these guys are associated with like raw oysters. Typically eat them raw. But the warmer the water gets, the more likely you're going to see infections associated with these. Freshwater, we're going to have Giardia lamblia. That's just one you can, like what they call travelers. It's like your hiker, right? You boil your water, but you still get sick. Giardia. And you'll be sick with what they call malodorous and greasy stools for months. Diarrhea. Hey, okay. So don't do that. Always treat your water properly. Naegleria fowler, I think you can boil it out. I think it is, you can kill it with boiling, but just always be sure, even if it's clear water that came from a beautiful waterfall, there could be a dead animal up above it. So boil, treat your water. Naegleria fowleri, this is the brain-eating amoeba. It's a 2% survival chance, even with treatment. Cryptosporidium, this is a protozoan that it can be found, it causes diarrhea, and it's resistant to chlorine. So where would you find it? Swimming pools. We see this outbreaks in swimming pools, public pools typically. Entamoeba histolytica, it's an amoeba. It's not brain eating. This one just causes diarrhea. And we have schistosoma mansoni. These guys get in, these are little, they're flukes. They're called flukes, and they're a type of parasitic worm, basically, flatworm, and they get into your body to infect you by going in through your intact skin on your body anywhere. So it causes what they call swimmer's itch. And then they burrow in through your skin and they get into your body, and they can cause serious health problems, liver issues, and things like that. Schistosomes. Then we have water treatment issues. The whole reason that we treat our water and what we test for when we're testing water, it's usually associated with fecal transmission, right? So cholera, Vibrio cholerae. For those of you who don't know, cholera is rice water stools. So it literally is like your diarrhea turns into like rice water. So white, so kind of your electrolytes basically are coming out of your body. It's causing your body to leak its electrolytes and its fluids in mass quantities. It can be extremely deadly. In some outbreaks, we've seen up to 70% mortality rate just from the dehydration that it causes. So it just depends, but yeah. Escherichia coli, E. coli, Shigella causes dysentery. Dysentery is bloody diarrhea. Salmonella, right. Hepatitis A can be found in the water, and usually is fecal oral transmission. Please get vaccinated if you haven't. And then Tinea solium. This is tapeworm. So we treat our water to protect from these things. Global warming is associated with higher risk for some of these outbreaks of the marine and freshwater diseases. And if you, I mean, I don't know what to tell you. Global warming, man, it's a thing. In the air, many labs and hospitals will test colony-forming units, which are like individual cells that can form a colony on a solid surface when you grow it. So colonies, those little bumps that you see on those plates, they come from one cell, just multiplying out that one cell. That's pretty impressive, right? Considering you don't see it when you first put it into the plate. Yeah, these guys are really, really tiny. So anyways, many labs and hospitals will test for that from their air to make sure their HEPA filters and stuff are working. So they'll just take plates that are auger plates like you are used to seeing, and just set them out and let them sit there for like an hour, and then send them to the micro lab to get analyzed. You can transfer things through the air by coughing, sneezing, excavation or construction, aerosolized liquids and droplets, disturbance of dust and dried particles, air conditioners, vents, and the wind. Basically, if it can move the air, you can transmit microbes that way, and therefore pathogens too. And they don't flourish in the air because there's no nutrients there. It's harsh environment otherwise, but they can float around for quite a while, in some cases up to 8 months. So think about that when somebody who has tuberculosis was in the room before you. Common airborne pathogens include SARS-CoV-2 and SARS, it's not meant to be CoV, that's coronavirus, right? So SARS-CoV-2 coronavirus and SARS-CoV coronavirus. These are completely different diseases. They are now. SARS-CoV-2, that's COVID-19, okay? SARS-CoV, that's SARS. SARS is a different disease than COVID. Yes, it is. I mean, I'm just telling you. I don't know why people get confused about it. I'm just telling you that they're different because they are. They cause completely different things. SARS came naturally from the environment, and I think we are all in agreement from where COVID came from. So, Mycobacterium tuberculosis, it's obviously a bacteria, causes tuberculosis. You might think like, oh, well, that's not something that we really see here in the United States, but you're absolutely wrong. If you've worked in any ERs, and you know that it's quite rampant within the homeless population. And anybody who has is homeless population with HIV, very likely to have tuberculosis, very likely. Now, you might say it's a small population of people, but if you look at the whole world, worldwide, we have about 1/3, almost 1/3 of the population of the world is infected with tuberculosis. How about that? You think it's an old world problem? Influenza, flu, rhinovirus, cold, adenovirus, cold. There's about 200 different rhinoviruses alone that cause the cold. And all these different adenoviruses, all these different coronaviruses. We have RSV. They all fall into that cold category. So it's hard to make a vaccine against the cold because we have all these different viruses that can cause it. Bordetella pertussis. This is pertussis, or you can also call it whooping cough. Very serious in little infants, even in the United States still. So be sure that your vaccines are up to date. This is a vaccine that pretty much everybody gets. If you get tetanus, then you're getting tetanus vaccine. Then you're getting the vaccine for pertussis, typically. All right, so we keep talking about bacteria growing on media. Medium is the singular for that. So this is a liquid. We would call that a broth if it was liquid. Semi-solid, that's just literally like it is on the, it's just soft. Usually we use the soft ones for determining motility, but we'll talk about that more later on in the course. Don't worry about that right now. Or a solid substance, right? Plates, broths, slants, and butts. Plates, slants, always solid. Butts are often semi-solid, but they can be solid. They're literally like a tube, like a regular old test tube. Let me actually draw it. They're a test tube, and this, so it's filled with stuff. So here's the little lid on the test tube, whatever. And then this stuff here, if it's solid or semi-solid, that's a butt. Otherwise, it's a broth, it's liquid, right? Must be sterile to avoid interference. We'll use that autoclave. Once again, we're preparing it. In the next lab with aseptic technique, we'll discuss how to transfer the sample to and from media. But what even is media and how is it made? Oh man. Let's see. It doesn't want me to play. It's like adamant that I not play this. It's just not having it. Okay. So I tried it on the last one too. And I've tried it on like my screen, the presenter screen or whatever. I can't get it to work, but it's whatever. It's not that important. It's not like this is the life-changing video of the whole semester or something. No, I was going to show you a video of them making media. They basically take powdered forms of nutrients and mix them with some water, make sure that it's completely mixed, and with some agar. Agar is some acidifying agent that comes from algae. So you can have like a solid, nutritious version of things that bacteria would grow on, right? Anyways, and you can add different things to the media for some things to grow and some things don't. And we'll talk about those differences a little bit later in the course, but just so you're aware, that's a thing that exists. I think it talks about it a little bit in chapter 3, if you haven't gotten through chapter 3. Anyways, then you would, after it's all liquefied, put it into the autoclave to sterilize it. And once it has cooled down enough that it won't melt the plastic of that Petri dish that they're pouring it into, because it's just plastic, they will, it's cool enough, they'll pour it into individual plates. It's usually estimated, but you can pipette it out and get exact volumes too, but usually estimate. Then let it cool until it's solid, and then you can use it right after that. We will be using all different kinds of media in the lab. You won't have to make it though. I have my lab coordinator to do that. I just thought it would be nice to have you appreciate how that's done. I just don't understand why it doesn't work. Whatever. All right, so that's what we would have done. We would have used swabs, like basically cotton swabs like you would to clean your ears or whatever, but sterile. So we'd streak it on, the bench tops or the handles of the drawers or your phones maybe, or the bottoms of your shoes, whatever you chose. And we would have streaked it onto a plate to see what we could have gotten to grow, and then placed it in an incubator. An incubator is an environment that is warmer so that the bacteria are happier and they'll grow faster. incubator. Our incubator looks exactly like a refrigerator. We have 2 of them that look exactly like old, like 90s refrigerators. And everybody always tells me, like, I have so many students in the semester who will be like, oh, do we put this in the refrigerator now? And I'm just like, I get sick of saying it's not a refrigerator. But it's not. It is an incubator because it is warm. It's 33 degrees Celsius, not Fahrenheit. So it's 33 degrees Celsius. That is 91 degrees Fahrenheit. And so for the summer, I actually like to turn it up even warmer, about 37 degrees Celsius. That's 98.6. So body temperature. So a lot of them grow really well at that. So we can get it done faster in the summer. So usually it's every other day that we have micro, but in the summer we do every day. All right, so always come prepared, having completed your pre-lab, which a lot of us are getting access to, not everybody, but it seems, but a lot of us are getting access to Top Hat now. Be sure you cannot access Top Hat from a website. You cannot access Top Hat the first time. You cannot access it from a website that you Googled. Don't try to do that. Don't try to access it from your app that you downloaded for Top Hat. The first time has to be through that course materials area, just for you guys who haven't tried it yet, to save you the trouble. It has to be through there. Otherwise, it will just say all the time that there's not a course. Sorry, but yeah, it has to go through there. That's how it's linked through what you paid for. So that way you don't have to pay for it again. But yeah, we'll work on getting it working, even if we can't for certain individuals. We'll get it eventually. So just hang in there, be patient. We're almost there. All right. Well, we've almost got Top Hat working. I have no idea when we're going to get you guys in the actual lab, but it's okay. In conclusion, always come prepared, do your pre-labs, wear your lab coat, wear closed-toed shoes. Inform me of any accidents, dispose of waste properly in the regular trash, the bio trash, or the sharps, as you would see fit. And if you aren't sure, you can always ask. Disinfect your bench tops when you get to lab and after you leave. Wash your hands, don't eat or drink in the lab. Ask questions. Microbes can be found anywhere. Not all microbes are pathogens, but some of them can be, and some of them can be found on your body and then become pathogens. I grind my teeth at night, and for some reason it's really made my front teeth hurt today. Some microbes can be pathogens or just part of your normal biota, the normal makeup of your body. A shift can happen anytime that you have like a change of location, like what we see with E. coli. When you have a UTI, it's your own E. coli from your own booty. And so you get your infection. Almost every single UTI is E. coli. Not all of them, but almost every single one of them. So you had something, usually, even if you have like a yeast infection or something like that, this can be affecting it, or vice versa. You can take antibiotics to get rid of your bacterial UTI, and then that can lead to a yeast infection. It just kind of depends, but in the case of this, it's usually, it's not like you get a yeast infection from sexual activity, but sexual activity makes you more prone to having a yeast infection. Just because there's a lot going on down there, right? So proper cleanup, always pee after sex and all of that is all I can say. That's the rules, and they're there for a reason. So do the best you can. Women are more prone to UTIs because they have smaller, shorter urethras. So men can still get them though. And then this is a UTI. UTI is a UTI. Media is used to grow microbes. Yeah. Okay, cool. So we've got some review of our topics, and I'm going to go through these, so don't worry about trying to answer them for yourself. I'm just briefly going to run through it and talk about it with you. So think about it as we're going through it. Yeah. Where would your paper towels used to wipe your disinfectant on your bench before starting lab get thrown away? So you wiped and cleaned your bench right before you were starting lab. It's disinfectant, it's paper towel, it's probably nothing else. And you don't have any reason to think it literally is. You don't see a spill from something. It's not actually contaminated with bacteria that you know of. Regular trash. Why should Betty be turned away for showing up to lab in strappy sandals? What if somebody, drops a tube and the glass gets stuck in her sandal and she cuts herself, or she gets bacteria all over her shoe or something like that, right? This is a safety issue, as well as trying not to bring that stuff home. Jordan dropped a test tube, cleaned it all up as best as he could, but he didn't tell the instructor. Why is this a problem? He could have left a piece of glass that somebody else will now step on. He could have left a slippery area of liquid that somebody else can slip and fall on. So just, there's all sorts of things that can go wrong with it. Just let me know, man. If you spill something, just be like, hey, professor, I spilled this over here, and I'll ask you if you can handle it. And that's usually how it goes, is that. So just let me know. Just, yeah, just be honest. Microbial level, what could be a problem with plastic pollution in our oceans? With plastic pollution, you have like water bottles and crap like that floating on the top of the ocean. And obviously, of course, anything that's gonna be floating on top of water and sitting out in the sun, and collecting just other plastic and other junk that's already on there and whatever is gonna be growing stuff on it and in it. So this brings new bacteria that aren't belonging in that environment into the environment, and now they're growing there. We're bringing more amounts of bacteria than should be there, even if they are the correct kind of bacteria. And this can lead to, we can have the plastic aggregating just with itself for those reasons, but also these microbes that are growing and creating these sort of films and everything like that can cause these islands to stick together more. And then that will lead to these development of these islands of plastic. There's one the size of Texas in the Indian Ocean. That's disgusting. Anyways, you can see how these could block out the sun in areas lower, deeper down where you might need like plankton, phytoplankton and stuff like that. They need the sun in order to live. Now you're blocking out the light in those areas for the size of Texas. It's not good. So that's the problem with that. Where do pathogens come from physically? Where did it come from though? Like originally, it came from other people's bodies or other organisms' bodies. It came from the air that was carrying it from something else. It came from the soil. It came from the water. That's all that's talking about. Okay. Why can't microbes thrive in the air? Because there's not enough nutrients and it's kind of a harsh environment. And then which would you expect to have more growth? A swab from the bottom of your shoe or a swab of a typical public toilet seat, and why? We'll get to that one in just a sec. This is our rundown of our answers. I will post all of this online. Again, this isn't like a questions that you're going to get on an exam or anything. But if it does help you understand the concepts, please, come back and reference it if you want to. For that last question, you're going to have a lot more growth from the bottom of the shoe because public toilets tend to get cleaned pretty regularly, several times a day. And not to mention it's a hard surface, typically on a toilet seat. So there's not like crevices or cracks for bacteria or other microbes to hide in. It's not very friendly for them to grow. Most people sit on a toilet seat with the flesh of their booty, not with, their mucous membranes and stuff. Most people. So you're a lot less likely to find bacteria in those parts of the body. I mean, you might see some, but it's not like it's your fecal matter everywhere all the time. It's not really how it works. There's some splashback from these toilets, but there's quite a lot of power involved to help rinse that stuff down in public toilets, typically. And like I said, they're regularly cleaned. How about you walking around in those shoes in that bathroom? Did you wash your shoes after you walked in that bathroom? They probably don't clean the floor as well as they do the toilets. I mean, so you're walking around with that. Maybe you stepped in dog poop. Did you really clean your shoe after that? Maybe you stepped in and you didn't know. What about the bird poop that you stepped in? What about the dirt? What about the, anyways, shoes are way grosser typically. So think about that the next time you walk into your house with your shoes on. Not that I don't, I do, but just saying, something to think about. All right, so that's day 2. Tomorrow, so everything's gonna be on Zoom for the rest of this week for sure. I just wouldn't, if they told me that the lab was open tomorrow, I don't have time to grow bacteria by then. They took my lab away before I was able to culture any of the stuff that you guys are going to be working with, let alone your cultures. So I don't have anything grown. So there's no way that we're going to be working in the lab this week. There just isn't. So for sure, tomorrow is going to be on Zoom again. I apologize. If I think that some of these other ones, like the stains that we're gonna be getting into are gonna be better served like in person, even if we can't do them in person, at least like having a demonstration in person or something. If I can get the materials, then we might try that. But I will let you know that won't be until next week though. So for this week, all Zoom. Okay, tomorrow, Wednesday, Zoom. Thursday, Zoom. Okay. And that's that. Does anybody have any questions other than that? Did Rachel ever get on? I didn't see. Let's see here. Okay. All right. Well, that's that. If Top Hat is working, then no, we're going to keep pushing ahead. It makes absolutely no sense with the format of this course to push the Top Hat assignments. They go with that lab. So no, yeah, we'll just have to do them for the time being with our talk. I mean, you would have to have them done by now anyways, right? So that's the idea. So we're sticking with that. If you can't get on Top Hat, then I will push yours individually, but it will not be everybody's. And you'll have to like kind of prove it to me that you can get on Top Hat. So just saying. Yeah. So do your best to get on and access that because I'll be checking that now that I have seen that it seems to be working for some people. And we'll work on people that it isn't working for and get that going. But yeah, I can't, it's just because the summer it's too short. I just can't have all that building up all at once. It's going to be too much work for you guys. But yeah, we'll be working through how to fill out those assignments though. So don't worry. Like, it's not like you have your 5 questions or whatever on this first one has more points to it, but normally you'll have 5 questions associated with your pre-lab. And that's it. And so, being familiar with the content is really all that's about. And then we'll talk about the content in detail. And then the post lab or in lab, they're the same thing, is the stuff that we got from the results. And if we don't have results, like tomorrow we won't have results, then we'll do something else and I'll let you guys know what it needs to be to upload it. Okay. So we'll just look at it that way. I'll walk you through it all. So we get it all done together. And so you know what you need to get done. I promise you it's not as much work as you think it is. A lot of this stuff is just knock it out and just have it done. So yeah. Anyways, any other questions? All right. I do see Keegan, Felicity, and Madison and Carlos already signed up on here. So I'll be sure everybody else that you get to it when you can. So I'm gonna go ahead and kick everybody off here. If that's all the questions that we have. And yeah. Cool. All right.